Bismillahirrahmanirrahim. Dear students, this is Inorganic Material Chemistry Chem 3115 and this is second lecture and we will discuss Introduction to Nanomaterials. The key concepts of this lecture includes nanomaterials introduction, their classification based on dimensionality, morphology and composition. In this lecture, I will talk about introduction to nanoscale materials, which are also called nanoparticles or nanomaterials. Moreover, I will talk about their classification, which will help us to understand the basic concept of nanomaterials. The question is, nanomaterials, what is the concept of nanomaterials? According to Nobel laureate Richard Feynman, who gave a visionary lecture, and in his visionary lecture he says that there is plenty of room at the bottom. What does it mean? He gave the concept of nanotechnology at that time when the researchers were not working on nanomaterials. According to Richard Feynman, he says that what I want to talk about is the problem of manipulating and controlling things on a small scale. What does it mean that manipulating and controlling things on small scale? According to Richard Feynman, the materials can be grown into smaller and smaller size until we reach atomic or molecular level. So this is the plenty of room at the bottom. It means if we move from microscopic size to small size, we can move to atomic or molecular level, which is far smaller than nanomaterials. The term nano is derived from Greek word nanos, which is used for dwarf. Dwarf means in daily life we can use this word for those people who are shorter in their length. This etymology and its placement on the metric scale, 1 nanometer is equal to 10 raised to power minus 9 meter, shows that tiny dimensions not visible to naked eye beyond the normal limits of our observation. It is well known that the diameter of hair is 100 micrometer. If we compare the diameter of hair, which is 100 micrometer, to the Buckminster fullerene, which is 1 nanometer in size, and it has been shown on next slide in figure 1.1. Nanomaterials are defined as materials having at least one dimension in the 10 to 100 nanometer range, in which nanoparticles are the major investigated subjects. The term nanoparticles was first appeared in 1980s. This is figure 1.1. This figure explains some materials and some particles. This is the length scale for classifying nanomaterials. On the right hand side of the figure, we can see diameter of a hair. It is 100 micrometer. From here to red blood cells, we can see there is a decrease in size. The red blood cells are in the range of 10 micrometer size. Escherichia coli is 1 micrometer in size, and we can see HI viruses, they are only 100 nanometer. A molecule of hemoglobin is 10 nanometer in size, and Buckminster fullerene, which is a carbon compound, 60 carbon atoms are interconnected to forming a cage structure. Its size is 1 nanometer, which I have mentioned on previous slide. If we compare the diameter of hair to Buckminster fullerene, which is made of 60 carbon atoms and its size is 1 nanometer. We can also find nanoparticles or nanomaterials in the history. In this slide on the right hand side, this figure, it is actually ancient stained glass. Many people in the history, they have been using different metals like gold, silver and other previous to stain different architectures. At that time, people did not know that what they were using, the gold materials or silver materials, their solutions, their colloidals, they were actually in nano size. Later on, when characterization techniques have been developed like scanning electron microscopy, transmission electron microscopy, and when these pictures were analyzed on high resolution, it was found that the pictures are made of small dots, just like yellow dot, blue dot. These dots are made of very tiny particles of gold, silver and other. There is a question, nanoparticle or nanomaterial. Sometimes when we read the literature, we can see both of the terms nanoparticle or nanomaterials. Here are some definitions by standards to define these terms given in the table. These are the organizations just like ISO International Standard Organization, ASTM, NIOSH, SCCP and some others. These are the definitions according to these organizations. We can see for a nanoparticle, according to ISO, a particle spanning 1 to 100 nanometer diameter, an ultraphy particle whose length in 2 or 3 places in 1 to 100 nanometer. This is some other language other than English. A particle with diameter between 1 and 100 nanometer, at least one side is in the nanoscale range, and on the other hand, material for which at least one size or internal structure is in the nanoscale, or material for which at least one size or internal structure is in the nanoscale. Material consisting of a nanostructure or a nano substance. So all those particles or materials which are 1 to 100 nanometer or their one or more than one dimensions are in the range of 100 nanometer, they are called nanoparticles or nanomaterials. While talking about nanoparticles or nanomaterials, we can also discuss about nanoorganisms. Because this is the lecture about bio-nanotechnology, so we will discuss about some biomaterials which are naturally occurring in nanoscale. Nanoscale organisms, commonly known as nanoorganisms, are found all around us and even inside our bodies. The category nanoorganisms are naturally occurring nanomaterials that include a massive range of organisms. For example, nanobacteria, viruses, as well as fungi, algae, and yeast that can produce nanoparticles in their bodies. For example, viruses. Viruses are the largest structurally characterized molecular assemblies which are known to date, which can be a non-living crystal and a living organism inside host cells. Generally, they are considered to be harmful as they cause disease in bacteria, in plants, in animals, and in human bodies. Advances in molecular biology have increased the possibility to genetically tailor viruses for use as catalysts and biomaterials. Nano size, monodispersity, distinct shapes, selective permeability to smaller molecules, composition controlled by the genome, manipulation, self-assembly, and polyvalent rapid growth and stability towards pH and temperature are the properties that make viruses a unique category among nanomaterials. The virus nanoparticles, as shown in this figure, they can be prepared by using different processes. From these nanoparticles can be prepared from viruses by removing their genetic material and making them nano cages for targeted drug delivery. So this is very good property of the viruses, that they can be used as drug delivery. Next, the second one are nanobacteria and nanobacteria and nanobes. Generally, bacteria will bind to soluble toxic heavy metals and precipitate them to their surface, producing metal nanoparticles. These are called nanobacteria, which are highly useful in the biosynthesis of low toxicity nanoparticles. So many metal nanoparticles such as gold alloy nanoparticles, uh, similarly non-magnetic oxide nanoparticles, and metal sulfide quantum dots such as cadmium sulfide and zinc sulfide, which are very common in inorganic chemistry, were synthesized using different strains of bacteria. The third one are magnetotactic bacteria. Magnetotactic bacteria are highly helpful to produce magnetic oxide nanoparticles that possess unique properties such as supermagnetism, high corrosive force, and microconfiguration, which can be utilized for biological separation and in biomedicine field. Generally, biocompatible magnetides like iron oxide, iron sulfides, and uh, and magmides, they are synthesized using magnetobacteria, are helpful in targeted cancer treatment via magnetic hyperthermia. The fourth one are algae, fungi, yeast, and bacterial spores. Algae supports the formation of silver nanoparticles, cadmium sulfide nanoparticles. Since very limited studies are available on algae, fungi, yeast, and bacterial, so the possible mechanism for algae-mediated nanoparticle formation are still unidentified. Similarly, fungi are utilized for the synthesis of nanoparticles, and literature suggested that they are excellent candidates for metal and metal sulfide nanoparticle synthesis. Next is the nanoparticles and nanostructures in the human body. The human body consists of nanostructures without which normal function of the body is impossible. It is formed by nanostructures such as bones, enzymes, proteins, antibodies, and DNA. A list of nanostructures that exist in the human body is presented in table. I will show you just later. Even some works categorize bone as a nanomaterial comprised of hierarchical inorganic, nanohydroxy, peptide, and organic collagen. Additionally, microorganisms such as viruses and bacteria are nanostructures that can cause diseases in humans. This is the table which list with where is the list of nanostructured particles associated with the human body. These are the nanostructured particles and this is their size. We can see the glucose, its size is 1 nanometer, so it means glucose is present in simple molecular form in our body. DNA, its size is 2.2 to 2.6 nanometer. Average size of protein, Ruisco monomer, this is the monomer, the simplest protein, it is 3 to 6 nanometer in size. Hemoglobin size is 6.5 nanometer. Sometimes it is said it is 10 nanometer in size. Micelle, it is also a special kind of organelle. Its size is 13 nanometer. Ribosomes are present in our cells. These are round structures. They are 25 nanometer in size. And similarly, enzymes and antibodies, they are present in our bodies. They range from 2 to 200 nanometer in size. Nanoparticles and nanostructures are also present in plants. Plants are also living organisms and we are talking about biomaterials. So as an introduction about nanoparticles, we can discuss some nanoparticles and nanostructures in plants. Wood is made up of natural fibers that are considered as cellulose hierarchical biocomponents. We know that wood is made of natural fiber and this fiber is made up of cellulose material. Cellulose is a carbohydrate. Natural fibers are composites of cellulosic
fibrils at the nanoscale level. In other words, we can say that the cellulosic fibrils, which are nano scale materials, they combine with each other to form composites which are natural fibers. The simplest form of nanometer-sized cellulosic fibers are 100 to 1000 nanometer long, containing both crystalline and amorphous segments. So, cellulosic fibers have two kind of segments, crystalline and amorphous segments. What are crystalline and amorphous segments? The students can learn about it from crystalline and amorphous solids lecture in BS Chemistry 6th semester. The unique strength and extreme performance properties of various natural fibers such as wood are attributed to their elementary hierarchical structure with nano fibrillar components. The isolation of nanocellulose from natural source is possible through nanotechnology. So we can use nanotechnology to isolate nanocellulose from natural sources, which requires combined methodologies including mechanical, chemical, and other processes. The resulting cellulose nanofibers would have distinct morphologies such as rod-like nanoparticles, or they can be entangled networks.
On particular importance, the optical property is one of the fundamental attractions and a characteristics of a nanoparticle. For example, a 20 mm gold nanoparticle has a characteristic wine red color. A silver nanoparticle is yellowish gray. Platinum and palladium nanoparticles are black. Not surprisingly, the optical characteristics of nanoparticles have been used from time immemorial in sculptures. And paintings. Even before the 4th century AD, the most famous example, which is the luxurious cup you can see in the figure, 4th century AD, illustrated in figure 1.1. This extraordinary cup is the only complete historic example of a very special type of glass known as dichroic glass that changes color when held up to the light. The opaque green cup turns to a glowing translucent red, just like here it is shown the figure A and the figure B. In figure A it is green cup and it turns to a glowing red color when light is shone through it internally. In other words, light is incident on the cup at 90° at the viewing direction. Analysis of the glass revealed that it contains a very small quantity of tiny, about 70 mm metal crystals of silver and gold in an approximate Mohr ratio 14:1, which give it these unusual optical properties. It is the presence of these nanocrystals that gives the luxurious cup its special color display. Until the Middle Age, the reputation of soluble gold was based mostly on its fabulous curative powers of various diseases. For example, heart and venereal diseases, dysentery, epilepsy, and tumors. It was also used in the diagnosis of syphilis. The history of the nanoparticle from ancient times of the middle from ancient times to the Middle Ages has been summarized by Daniel and Astruc. The first book on colloidal gold was published in 1618 by the philosopher and medical doctor Francisci Antoni. This book includes considerable information on the formation of colloidal gold sols and their medical uses, including successful practical cases. The book noted that the soluble gold appeared around the 5th or 4th century before Christian in Egypt and China. On the other hand, industrial manufacturing of stained glass with colloidal particles was established by Kunkel in the 17th century, about 1676. He also published a book whose chapter number 7 was concerned with drinkable gold, that contains metallic gold in a neutral, slightly pink solution that exerts curative properties for several diseases. He concluded that gold must be present in aqueous gold solutions to a degree of contamination such that it is not visible to the human eye. A colorant in glasses, that is the purple of Cassius, was a colloid resulting from the presence of gold particles and tin oxide, and was highly popular in the 17th century. A complete treatise on colloidal gold was published in 1718 by Helcher. In the treatise, this philosopher and doctor stated that use of boiled starch in its drinkable gold preparation noticeably enhanced its stability. These ideas were common in the 18th century, as indicated in French Chemical Dictionary, which is dated 1769, under the heating or potable. It was said that drinkable gold contained gold in its elementary form, albeit under extreme subdivision suspended in a liquid. In 1794, Fahlem reported in a book that she had dyed here it means coloring, that she had colored silk with colloidal gold. In 1818, Jeremias suggested an explanation for the differences in color shown by various preparations of drinkable pink or purple gold solutions, in that these solutions contained gold in the finest degree of subdivision, whereas yellow solutions were found when the fine particles had aggregated. In 1857, in a well-known publication, Michael Faraday reported the formation of a deep red solution of colloidal gold by reduction of an aqueous solution of chloroaurate AuCl4 by phosphorus in cadmium sulfide. Here is the chronological table of nanotechnology, which shows a complete history and development in the field of nanotechnology. Starting from 1200 to 1300, discovery of soluble gold. I have just talked about this history, and now I will again explain from this table. From 1200 to 1300 before Christian, discovery of soluble gold was done, and it was occurred in Egypt and China. Then 290 to 325, Lycurgus cup, which was discovered in Rome, which was invented in Rome. 1680, first book published on colloidal gold by F. Amonti.
1676, Germesius published book on drinkable gold that contains metallic gold in neutral media. In 1718, publication of a complete treatise on gold colloid. 1857, it was the first time when Michael Faraday synthesized colloidal gold. In 1902, surface plasma resonance was discovered. In this history, we will also see the different instruments which have been used for the analysis of nanoparticles, for the characterization of nanoparticles. Like in 1908, scattering and absorption of electromagnetic fields by nanospheres, it was happened. Then in 1931, transmission electron microscope was discovered, and in 1937, scanning electron microscope was discovered. 1959, Feynman Feynman laureate, which was a Nobel Prize laureate, and I have discussed about it in this lecture. He gave the concept of plenty of room at the bottom, which means that the materials can be grown into smaller and smaller into the size, and we can reach to atomic level. Then 1960, microelectrochemical systems, mechanical systems were developed, and 1960, successful oscillation of the laser was carried out. Then 1962, the Kobo effect. It is a very famous effect which was presented by Kobo, University of Tokyo. Similarly, Moore's law, the Honda Fujishima effect, all of these effects, they were concerned with nanotechnology. And 1972, amorphous heterostructure procedures were developed. And in 1974, the concept of nanotechnology was proposed by Tokyo University of Science. This is a complete history of nanotechnology.
The nanoparticles are so small that if we take a cross section of a hair, 50,000 nanoparticles can accommodate on the cross section of the hair. I can also ask you a question. Have you ever seen nanoparticles in your daily life? We are talking about nanomaterials whose diameter is 1 to 100 nanometer, and this diameter is even smaller than the wavelength of the visible light, which is from 400 to 700, and in some books from 350 to 750 nanometer. So when we compare the diameter of the nanoparticles, which is very small, it means that we cannot see these materials using our naked eye under visible light. However, the nanoparticles are always dispersed in the air in large quantity. If we see some sunlight which is coming from a window, you can see some particles are illuminating. Or if you stand in front of a multimedia, you can see some particles are blowing in the air. So these are the nanoparticles. Next is the classification of nanomaterials. They can be classified depending upon their dimensionality, morphology, and chemical composition. So starting from dimensionality, the nanomaterials can be classified into 0-dimensional, 1-dimensional, 2-dimensional, and 3-dimensional nanomaterials. 0-dimensional nanomaterials are those materials which are in nanometer scale in all dimensions. 1-dimensional nanomaterials which are with one dimension not in the nanometer scale, they are typically thin films or surface coatings. So 1-dimensional nanomaterials are in nanometer scale along 2 dimensions. 2-dimensional nanomaterials are those nanomaterials which have 2 dimensions not in the nanometer scale. It means the third dimension is in nanometer scale. 2-dimensional nanometers include nanotubes, dendrimers, nanowires, fibers, and fibrils. 3-dimensional nanomaterials are those materials which are not confined to the nanoscale. in any dimension. These materials are thus characterized by having three arbitrary dimensions above 100 nanometer. Here are some examples of zero-dimensional nanomaterials. So these nanomaterials are those nanomaterials in which all of the dimensions are in nanometer scale. In one-dimensional nanomaterials, the two dimensions are in nanometer scale and the third one is out of nanometer scale. In two-dimensional nanomaterials, the two dimensions are out of nanometer scale and only one dimension is in nanometer scale, and the complex structures which are three-dimensional nanomaterial structures, they are not in nanometer scale along any of the dimension. Their morphology. The morphological characteristics to be taken into account are the flatness, aspect ratio, and spatial position of each element in the case of hybrid nanoparticles. A general classification exists between high and low aspect ratio particles. High aspect ratio nanoparticles include nanotubes, nanowires. On the other hand, small aspect ratio nanoparticles include spherical shaped, oval shaped, cubic shaped, prismatic, helical, and pillar shaped nanoparticles. Transmission electron microscopy can be used to see the exact morphology of the nanoparticles. As an example, the TEM images of some nanomaterials with different morphologies are given on the next page. These are different morphologies of the nanoparticles which have been studied by using transmission electron microscopy. In this figure 1.4, the A figure, it is monodispersed copper nanoparticles. These particles are very small in size. Figure B represents iron nanorods, and similarly in figure C it can be seen copper, silicon, core shell nanoparticles. Core shell nanoparticles will be described later. Similarly in figure D, porous iron oxide nanoparticles. Figure E is representing iron oxide cubes decorated with nickel nanoparticles. Figure F is representing porous silica spheres with alpha iron oxide nanoparticles which are embedded in pores of silicon oxide. Similarly, in figure G, gamma iron oxide nanoparticle spheres are shown. The third category of nanoparticles is the composition of the nanomaterials or nanoparticles. Nanoparticles can be divided into three types: mixed nanoparticles, core shell nanoparticles, or layered nanoparticles. Mixed nanoparticles can be either random or ordered. Figure 1.6, which is on the next page, it is showing mixed nanoparticles. Randomly mixed alloys correspond to solutions of solids, whereas ordered nanoparticles correspond to ordered arrangement of A and B atoms. The A and B atoms are shown in figure 1.6. Core shell nanoparticles consist of a shell of one type of atom, B, surrounding a core of another type of atoms, A. It is also represented on the next page. Layered nanoparticles are commonly referred to in the literature as genus nanoparticles. They consist of two types of nanoparticles, A and B, sharing a common interface. This is shown in figure 1.6c. These type of nanoparticles tend to minimize the number of bonds between elements A and B. This heterojunction structure facilitates phase separation. Here is the figure 1.6. The A shows mixed structure nanoparticles. We can see A and B atoms are mixed in the cluster structure. The atoms are randomly arranged among the structure of the nanoparticles. The figure B is representing core shell structure. In this structure, the B atoms are making a core shell and the A atoms are embedded in the core shell structure. This is second type of nanoparticles. The third one, figure C, is representing layered structure. In this structure we can see the A atoms, they are making one kind of layer, and B atoms, they are making second kind of layer. These atoms are continuous in A layer. Similarly, the atoms in B layer, they are also continuous. This is layered structure. The references for this lecture are There is Plenty of Room at the Bottom by Richard Feynman. His book published in 1999. The second one is Introduction to Nanoparticles by Satoshi Horikoshi and Nick Sarpon. So these two books can be downloaded from the internet. The assignments are students have to find more examples showing different type of nanoparticles depending upon their morphology and size. Students have to search the detailed history of nanoparticles and make a comprehensive report. The students should search different metal oxide nanoparticles examples and show their physical properties. Thank you very much.